In Lab 9, we will look at microbial diversity. In other words, the different kinds of microbes in the world around you. This includes bacteria, algae, protozoa, fungi, and viruses. The only thing you're going to need for this particular lab is something from your house that's made from a microbe, which I'll describe at the end of this lab. The lab begins with a continuation of studying the geological timescale and life's history on Earth. We will be exploring how life on Earth began, starting with the bacteria and the archaea. We will also do a refresher on the classification hierarchy. You should be able to list this in order for tests and quizzes. We will look at the tree of life with the 3 domains, bacteria, archaea, and eukarya. Here is a place for you to practice the classification hierarchy and for you to come up with a clever way or mnemonic to remember them in order. We will begin our study of microscopes because all of our microbes are so small you need a microscope to see them. There's also an opportunity for extra credit for a virtual lab on the microscope. So we'll look at the microscope first, and then as I scroll through here, we'll begin our investigation of bacteria. For each microbe, you'll look at characteristics, some beneficial or important facts, and then we're actually going to look at them. And since you don't have microscopes at home, you will be using the additional PowerPoint slides. They are found both in the lab manual and on the Canvas site. Our first microbe is going to be bacteria. We will look at the bacteria E. coli, which is found in our gut, and it is a good bacteria. So here you have an image of bacteria when viewed under the microscope. We have stained E. coli pink so that you can see it a little bit more clearly. This particular slide or image was used by using the 100x objective lens of the microscope and the 10x of the ocular. One of the things that we're asking you to do is draw what you see, so draw what you see in this picture, and give the total magnification. Remember, total magnification is objective lens times ocular lens, so that would be 1,000, because 100 times 10 is 1,000. Then as you continue to scroll, we'll look at a second bacteria, Streptococcus pyogenes, which is the bacteria that causes strep throat. So if you were to have your throat swabbed, and you looked at it under the microscope, if you were positive for strep throat, this is what you would see. Then we have a probiotic, which is going to introduce good or healthy bacteria into your system. As we continue to scroll, we have archaea, algae, protozoa, etc. So these are the slides, and you can even view some animations using YouTube you're going to use to complete the lab. So now we're back in the lab exercise. So you're going to draw E. coli, you're going to draw Streptococcus pyogenes, and you're going to put down the total magnification. You're going to fill in the 3 or 4 bacteria found on the label for the probiotic, and you're going to answer the questions. The answers to the questions are going to be found within the lab exercise or lab manual. This is photo 1. You're going to continue to do that for the remaining microbes, including algae and protozoa. So here we are at the protozoa. This is our next photo. So again, you're going to go to the PowerPoint presentation, draw an amoeba, draw a paramecium, draw euglena. Then you're going to click on this video. You're going to watch how amoeba moves, in comparison to paramecium and write your answer. For each of these, 22, 23, 24, you're going to circle how each of these moves and answer the remaining questions. This is photo 2. Then we get to fungi, and lastly to viruses. And again, you're going to read and fill in all of the information. Photo 3 is going to be you kind of taking all the information that you've learned and putting it together. And this is one, sometimes you skip a few steps. So here's what you have. You have the tree of life with our 3 domains, bacteria, archaea, and eukarya. The first thing I'm asking you to do, question 35, is to fill in the cell type. So for each of these, you're going to write either prokaryote or eukaryote on the line that says cell type. Then, using the examples in 36, you're going to fill in the appropriate number of lines. So for example, there'll be 3 different bacteria from the top. There'll be 1 archaea, which I'm actually going to give you a hint on that one, and then the rest will be eukarya. You are graded on accuracy, so make sure that you're positive about what goes where. All of the answers are in the lab manual. So for example, under archaea, our example is going to be Pyrolobus fumari. So you're going to put that on the line under example. Take a photo, it needs to be legible and clear so I can read your answers, and that is photo 3. Then you have a crossword for practice, which is just that, it's for practice. Those are great study aids for tests and quizzes. Then we get to the last photo. So this is the first of these that you're going to do. You'll do one with plants and animals in the next few weeks. So here's how this is going to work. You're going to start with an index card, put your name and the date. What else do you need? You need the name of the product. So you're going to find something in your house, and we're going to use an example of Swiss cheese that was made by bacteria. And you can't use Swiss cheese because that's what I'm using for an example. So something in your house that was made by a bacteria. There's lots of options. And the type of bacteria that made it. So on your index card, you're going to have your name, the date, the name of the product, so Swiss cheese. And in this case, bacteria, because that's what made the Swiss cheese. So you're going to hold up your index card and the product with your selfie. And that is going to be photo 4.